Hey, it's Mike Rowe, and it just occurred to me that ZipRecruiter has been sponsoring my podcast, The Way I Heard It, for the last 10 years. Consequently, I've agreed to sing their jingle at the end of this commercial in 4-part harmony, all by myself. Why would I do that? Because years ago, when the MicroWorks Foundation was expanding and I was desperate to hire a few uniquely qualified individuals, I posted a job for free on ZipRecruiter, and I found the people I was looking for in less than 24 hours. It's that simple. Ever since, I've been telling my audience that ZipRecruiter got me the most qualified, most interested people. And now I'm telling you. Post a job for free at ZipRecruiter.com/zip and watch what happens. That's ZipRecruiter.com/zip. And now, here's their official jingle in 4-part harmony. Build your team. Build it sooner. Meet your match at ZipRecruiter. Try it free.
Every team, every topic, everywhere. This is Believe.
Welcome to Unbiased Science,
where we bring scientific method to the madness.
We're your hosts, Dr. Jess Dyer
and Dr. Sarah Scheinman.
And today, we are talking about the P word, because it is all over our feeds. Of course, I am talking about peptides. Just to tee this up, so I usually do a little bit of a public health debrief, right? But I'll make it relevant to the topic at hand and can't wait to introduce you to our special guest. But before I do, here is a headline from NPR. This was last month. The wellness world is eager for RFK Jr.'s promised move on peptides. So this is huge. Peptides are huge in the wellness world. You have folks like Joe Rogan has gone on his podcast talking about how they've changed his life. And RFK Jr. also feels that they are, that they're very good for us, and has said that he signaled that he is expecting the FDA to change the status, basically to loosen some restrictions on peptides in the next couple of months. And we'll talk about this more, but there are some other things in the works with compounding pharmacies where you might be able to get peptides through those. So let me stop talking and let's introduce our very special guest, Dr. Leigh Baxt, who is an unbiased scientist. She is a regular contributor to Unbiased Science, yes, and has established herself as kind of the peptide queen on social media right now. So we are super, super fortunate to speak.
Peptide queen.
Oh God, I don't know if I want that. It just makes me think of that lady who's like the worm queen. I don't remember what her name. You know, the lady she's promoting parasite cleanses and says you have to like eat ice cream under a full moon to get your parasites to come out.
Oh, wow.
Yeah. She's like the worm queen or something. I don't know. I might not be remembering the name right, but
I'd rather be the peptide queen, honestly.
Yeah. I guess I like to think about it as I'm sort of secretly teaching you about drug development and discovery, which is what I do and what I love, but I'm using peptides as my case study. So it's like, it's a win-win, I'm teaching you about the processes that I do and why they're important in the context of something that mostly hasn't gone through those processes.
Amazing. Sarah, do you want to read a little or share a little bit about Leigh's background?
I would like to read a little bit about the peptide queen, aka Dr. Leigh Baxt, who is a translational and drug development scientist specializing in the interface between basic biology and therapeutic development. Her work focuses on advancing early-stage ideas from basic science through in vivo validation, generating rigorous decision-enabling data to determine which therapeutic concepts are robust enough to progress. She has contributed to more than 30 therapeutic programs across oncology, metabolic disease, infectious disease, and regenerative medicine, with experience spanning small molecules, biologics, and PROTACs, along with earlier stage work in drug conjugate modalities, including antibody drug conjugates, small molecule drug conjugates, and peptide drug conjugates. Leigh also teaches translational science to a range of audiences, mentors junior scientists, and shares science communication content on Instagram as a way to extend her teaching beyond the lab. Leigh, welcome to the show. We're very happy to have you.
I'm really excited to be here. You don't know this, Sarah, but Jess knows this. I have followed Unbiased Science for many, many years. And I used to message Jess all the time whenever I had like any useful things to add to her content or posts. And I've been a longtime follower and commenter and supporter and reposter. And then took the dive into the SciComm world like a year ago. And within like a month of doing that, Jess was like, hello. And obviously I said yes. And so being here on this podcast is very surreal for me.
This sounds like our Jess.
Oh,
yeah, absolutely.
Goodness. I have like warm and tingly feelings right now. I love it.
Well, you are great. You're great at bringing people together. I will just say that.
Oh, thank you for that. Guys, this is so sweet. Thank you. Okay, Sarah, I have a feeling I know what your first question for Leigh is going to be.
Okay, so you know how I like to set the stage. And we were just talking about how the P word, peptide, it's all over the place, but I feel like very few people actually know what we're talking about. So maybe let's kick things off with a definition, Leigh. What is a peptide?
It is a small piece of protein. It is just a small chain of amino acids. Lots of proteins are actually made from peptides when they are produced in our bodies. I think you teach biochemistry, right, Sarah?
I do, yes. So this is my understanding of peptides, but I am chronically off social media, so I actually didn't realize that peptides were such a big thing in the wellness space these days, because I just know them as chains of small chains of amino acids.
And that's what these are too. So like the first piece I put out about peptides was basically just saying that, peptide is really just a descriptor. A peptide isn't really like a specific thing. It's just a small piece of protein. Mm-hmm. And one of the things a lot of people don't realize is we actually have more than 100 FDA approved drugs that are in fact peptides. Insulin is a peptide. Mm-hmm. GLP-1 agonists are peptides. So the idea that peptides are some new thing that didn't exist before is just, I guess short-sighted, because peptides are obviously around all the time in our bodies, mm-hmm. and we have lots of drugs that are peptides too. Right. But right now there's sort of this, I don't know if I want to call it a craze, but there's a trend where there are
I call it a craze. I would call it a craze.
Okay. There are
craze, trend, potato.
Yeah, there are a number of peptides out there which are, small sequences of amino acids. Many of them are just fragments of larger peptides that are in fact produced in our bodies, but many of them don't actually exist in that specific form in our body, even though people often call them natural. Like BPC 157 is a fragment of a larger peptide. TB 500 is a fragment of a larger peptide. So the sequence itself, is part of that bigger peptide in our body, but these smaller peptides don't exist in their actual form as they are in our bodies. But people are touting these as having all these incredible properties that are like healing and, I don't know, like wellness, everything, anti-aging, healing from injuries, like boosting your energy.
Yep.
I've seen immune boosting. I've seen all kinds of claims. And what is disturbing is that the majority of those claims for these non-FDA approved peptides, so these are different categories, are based on animal data or cellular data, and there's very little human data. For some of them, there's no human data. For some of them, there's maybe like a tiny bit of human data that's sort of, small descriptive studies, nothing that's like a robust randomized clinical trial. And most of these, are not going through the drug development process. So it's kind of like the Wild West. I saw there was a physician who writes about this, and that's what he called it, the peptide Wild West.
That's the perfect term for it. I will say I've been a bit frustrated because I see that peptides are quite popular in certain wellness circles where we actually have high rates of vaccine hesitancy. And it's like these things, and I know we're going to get into it, but it's like these are so well tested, regulated, all of the safety and efficacy data. And then we, as you said, the Wild West of peptides, we don't have that for peptides, yet people are willing to inject this into these things into themselves. So that drives me bananas.
Yeah, I completely agree. It's hard to reconcile feeling that COVID vaccines or other vaccines don't have enough human data, that people feel comfortable using them, and they have critiques about how the human randomized clinical trials are run and how many, thousands of people are evaluated. But when it comes to these peptides, there aren't any RCTs, and you're also going to inject that. There aren't even regulations to ensure what's in the vial is in fact what you think it is, or that it's sterile. But those, for some reason, are okay to inject. And, the only thing I can say is I think because it's in this kind of wellness sphere, people don't view them as like a therapeutic. They think that they're sort of like enhancing. So because it's not a therapeutic or like a treatment for something, I don't know if the bar is different or just the way they think about it is different.
And well, Leigh, so you mentioned that there are some peptides that are pharmaceuticals that, as we know, are rigorously tested and undergo clinical trials.
Of course.
So what are the difference between like those kind of peptides and the peptides that you were mentioning that are maybe like being touted as like anti-aging or like that people are injecting without medical oversight? Where's the distinction there?
Data.
Yeah.
Data. So for something to be an FDA approved drug, it needs to go through, there's basic science where you're understanding the biology behind the pathway. So people identified GLP-1 receptor, they understood what the role was, they started finding compounds that showed these activities that were of interest. They figured out that activity went through a GLP-1 receptor and all of the signaling pathways. They then analogued peptides that were identified. I think some of them were even identified in the saliva of Gila monsters, which just tells you that like we have to fund basic science because you never know where things are going to come from.
You never know.
Right? So you optimize these things. You go through a development process to give them the best drug-like properties, meaning that they get to the right tissues, they have good stability, no toxicity, all of those things. So you go through an entire, let's say 5 years of preclinical development before you ask for approval to go into humans. And then once you have that approval, if you get it, and not everything gets approved, like the attrition rates in drug discovery and development are incredibly high. We can talk more about that later, but you get into the human development, you now still need to get through phases 1, 2, and 3, as well as FDA approval. So you have, a cumulative, 10 to 15 years of data that you have to provide to show evidence that something is safe and efficacious for it to be an approved drug. Most of these peptides, I would put in like not even quite into that preclinical space yet. We have like some basic science, some molecular pathways, maybe very tiny human studies. There are some where there's more human data, and it looked like maybe someone was trying to develop them in through like the correct regulatory framework, but then they were like abandoned because they didn't perform well. So, they're basically in like the first part of that process.
Hey, it's Mike Rowe, and it just occurred to me that ZipRecruiter has been sponsoring my podcast, The Way I Heard It, for the last 10 years. Consequently, I've agreed to sing their jingle at the end of this commercial in 4-part harmony, all by myself. Why would I do that? Because years ago, when the MicroWorks Foundation was expanding and I was desperate to hire a few uniquely qualified individuals, I posted a job for free on ZipRecruiter, and I found the people I was looking for in less than 24 hours. It's that simple. Ever since, I've been telling my audience that ZipRecruiter got me the most qualified, most interested people. And now I'm telling you. Post a job for free at ZipRecruiter.com/zip. And watch what happens. That's ZipRecruiter.com/zip. And now, here's their official jingle in 4-part harmony. Build your team. Build it sooner. Meet your match at ZipRecruiter. Try it free.
It's right now.
And correct me if I'm mistaken, the first part of this whole testing process, we focus mainly on safety at first, right? Before we shift to things like how effective. Is that right? You have to make sure.
In clinical or preclinical?
Clinical.
Yeah, clinical. Phase 1 is generally safety. And it's small. It's usually volunteers who don't have whatever, condition you're evaluating. They're just healthy volunteers, and you do like dose finding studies and you ensure safety. But the other thing that's important to emphasize is even when you move to phases 2 and 3, and even if you get something onto the market, there is safety monitoring that occurs throughout. You always look for adverse events. You always look for rare things. So it's not like safety ends after phase 1.
Right. And if I could actually just clarify a point that you brought up, that what do we mean by clinical and preclinical? The way that I understand it, and Leigh, correct me if I'm wrong, is when we're talking preclinical, basically what that means is non-human trials, like trials involving like the peptide in cells or in mouse models or something like that, which also involves looking at safety, right? Because if you're putting a drug in a mouse, you absolutely need to make sure, like, hey, is the mouse alive and thriving, or is the mouse dying because the dose is wrong? So that does involve some safety as well. But then when we're talking clinical trials, that involves humans, and then there are lots of different phases of that as well.
Yep, exactly. And not only safety in the preclinical space, there are specific safety studies that you have to do depending on the drug modality. So for small molecules, one of the things you'll hear about is something called safety toxicology and safety pharmacology. So for one of those, for the pharmacology study, you give kind of chronic dosing at a dose in an animal that you could extrapolate to like a human dose, right? So like a low level, whatever. And you look for specific types of adverse events over a long term. But then you also do toxicology studies where you actually give higher doses, or as high of a dose as you can see before you start to see adverse events. And there are rules from the FDA for what fold difference has to exist between the amount that you want to give versus the amount where you start to see any adverse events. So I have a friend working in a project right now who their margin for the indication they're developing for is 50-fold. So they have to show that their dose that shows the effect they want is 50-fold less than the minimal dose where you start to see any adverse event. So it's a pretty big threshold. Yeah, but even before that, which is sort of late preclinical, when you start a new drug development project, there's also safety in the target validation phase. So when you're thinking, is this a good drug target? Part of that evaluation is safety. So looking at animals lacking the gene to see whether that target causes any type of adverse events, looking for, human subjects who have mutations in the gene that produces the protein you're going to target. So safety is always part of it.
And Leigh, there are failure rates, right, in every phase of drug development. Can you talk a little bit about that?
Yeah. So I work at the very early stage, just moving from the idea to the very beginning of preclinical development. And I would say when you're looking at ideas that people have, like projects that they're proposing, and then you look at market approval, it's more than 99% failure. Yeah.
Whoa, that's a high number. Oof. Wow. Okie dokie. All right.
It's very high. So you're considering injecting something in your body that hasn't gone through any of that. That's why someone like me who sees these failure rates, you're like, maybe wait till we get a little more data. Like, I'm not anti, if you want to experiment on yourself, that's fine. But experimenting in a way where you have no quantifiable risk or benefit because we don't have any data, like that's past my comfort.
It's a little problematic, to say the least.
Well, Leigh, you just did a great post today, which was on social media, which was, I'm not anti-peptide, I'm just pro-data. And we just simply don't have the data yet.
Yeah.
I have a million questions, but I want to relay. We actually received a question. Maybe we could do a little Heard from the Herd segment. Basically, people are skeptical that we won't ever have these trials or data. Here, Leigh, do you know what I'm talking about? I'm going to pull up.
Because they think you can't patent it, so there's no money?
Yes.
Yeah, yes. So there are patents on a number of these peptides, actually. If you go look, like BPC 157, which is, I think, 15 amino acids, it's pretty small, so there's not a lot of space to kind of play with it and modify it, but there are still patents on it. So there is a rule that you cannot patent a naturally occurring, protein or protein sequence, and that is absolutely true. But what a lot of people don't understand is when you go through the preclinical phase, before you even have a candidate to nominate to put into humans, there's a lot of tweaking that occurs, whether you're changing the kinds of amino acids, whether you're adding some kind of side chain, whether you're going to cyclize the peptide. There's a lot of changes that occur. And this is true for biologics, for small molecules, and also for peptides. There are changes that you would make to give it better properties. We talked earlier about these drug-like properties. So things like, does it get to the right tissues? Does it stay in the bloodstream long enough to have the desired biological effect? A lot of peptides have very short half-lives, so you need to extend the half-life. That's part of the development for most of the peptides that are drugs. If you look at what changes were made and for what reason, a lot of them are stability and extension of half-life. So you go through all of these modifications. When you do that, you introduce enough changes that this is now no longer a, naturally occurring peptide sequence, and now you have what we call freedom to operate, which means you can patent it. So the ability to patent things is generally not, a super high bar for the pharmaceutical industry. Like, being able to patent something, there are people whose whole careers are focused around understanding that. And I actually spoke with someone who does literally make peptide patents with pharmaceutical companies. And I asked her, do you think that the reason we're not seeing a lot of companies diving into this space is because of patenting? Because I think it's because of data. And she said, no, I absolutely think it's because of data.
Ooh,
so, I mean, if the person who's the most expert in that field also thinks that, I don't know. I know it's one person. Obviously, this is anecdotal. But there's enough red flags that I would be concerned.
So let's talk a little bit about risks. Before we hit record, you were sharing a story about, I don't know, maybe you could just tell it now, but basically someone was affected by injecting peptides.
Yeah, I saw it shared. It was shared on Instagram by one of the physicians who I follow, and it was someone who had an infection with a bacteria called Mycobacterium abscessus, which is related to tuberculosis. It's a hard-to-treat infection. It can cause these really terrible skin infections, and I guess this person had been injecting peptides in their leg, and I don't know if they had poor sterile technique or if the peptide itself was contaminated in some way, but they had developed this terrible infection from their peptide use. So the lack of regulation for how these sort of gray market peptides are produced makes me worry about what you're actually getting.
And my question is, where are people even getting these from? I mean, I assume you can't go to a doctor and get them prescribed if they haven't undergone those clinical tests that we talked about.
So the scary thing is there are doctors who actually are selling peptide therapy and promoting peptide therapy. This morning I put a post on my page that was actually a snippet from the website of a doctor who, I'm not going to name names, but it was someone who was arguing with me about peptides and telling me there was all this human data. So I went to the website, and the peptide therapies that this person is recommending are in fact all these wellness peptides, and the sources that they cite on their website to say all these amazing properties they have are rat studies or animal studies or cellular studies. There's very little, in fact, no human studies at all listed for a number of those peptides. There are some where we do have some human data, which is great, and they're not FDA approved, but there's some data. There's others where people are either conflating two related things as if they're the same, or there just isn't data. So with the conflating, it's this peptide TB500. It's based on something called thymosin beta-4, which is a bigger peptide in our body. And there's lots of research on thymosin beta-4. But people will often say, oh, buy TB500, it does all these things. But that data is on thymosin beta-4. It's not on TB500. TB500 is a smaller piece of it. So it's a different molecule. It will behave completely differently. And I can give you a very interesting example about small changes later if you want, in terms of behavior.
No time like the present, or no?
All right, I'll tell you. So it's kind of an interesting story, actually. One of my colleagues, who's a medicinal chemist, shared it with me. We were talking about, how very small changes to structures can affect function. And there was actually a drug before ibuprofen. So many, many, many years ago, there was a drug called ibufenac. And the only difference between ibufenac and ibuprofen is a single methyl group, okay? A very small difference. Now, this is a small molecule, so it's a different story, right? It's much smaller. But ibufenac was released to the market. And this is before we had all of the regulatory strategies and the requirements for how we do clinical trials. All of those things have developed because of the issues we saw historically in drug development, which is why we have this framework of evidence that's required now. So ibufenat created this metabolite when it was processed by the body that was hepatotoxic. So it was bad for the liver. So they had all these people who were having liver damage from it. And so they had to pull it off the market, go back to the drawing board and analog around it. And they identified ibuprofen, which is just one methyl group different. And, you know, I took some yesterday.
Yes.
I take it quite a
So
bit.
small, you know, tiny changes can have really big impacts, which is why there's so much testing that you have to do when you develop new drugs.
So you mentioned regulation, right? And I know you're not a regulatory scientist, but can you say a little bit about, you know, potential changes? Again, as I mentioned, RFK Jr. has promised some regulatory changes. Like, what could that look like, and what would the next steps be?
So the way I understand it is that they would change the list of peptides that aren't allowed to be compounded would be changed, so that a number of these wellness peptides, like, you know, BPC 157, TB 500, would be moved from the not allowed to the allowed to be compounded list. There's two sides to this. Number one is by moving them to that list, there would be a regulatory framework applied for the manufacturing of those items. So people would be at less risk of getting things that are contaminated, that aren't sterile, that aren't actually what they say is in the vial. Keep in mind that that regulatory framework is still not near the level it would be required for the pharmaceutical industry. So, you know, take it with a grain of salt, but there would be some regulations. So in that sense, you're safer, right? And there's some evidence that will be required. I believe they're evaluating these in terms of their ability to affect certain diseases. But again, they're not being evaluated as FDA-approved drugs. So this is not an FDA drug approval process. The burden of evidence that would be required is nowhere near that level. So that's like the good side, right? More safety. The bad side is like, now all of these doctors who are already selling and promoting peptides, not all, like some, right? And other types of practitioners. Med spas are also a big one that I've heard about, that are selling these. You're going to see more of it. It's going to be more mainstream and more easily available. Doctors are going to prescribe them. You're going to have compounding pharmacies making them, stacking them. And this is another issue. When you make a drug that has multiple things in it, you have to study that in the preclinical and the clinical phase. There's a lot of analytical work that goes into that.
Because of the interactions.
None of that.
Right?
Exactly. There was a paper recently from, I believe it was Eli Lilly, where they took a bunch of I forget which their GLP-1 agonist is, but compounding pharmacies are taking knockoffs of their GLP-1 agonist and mixing them with vitamin B12 and selling that as some, like, you know, wellness benefit, whatever. So people are using it. And Lilly got a bunch of these samples and analyzed them and found that when you do the analytical evaluation, there are changes occurring to the molecules from them being together in that buffer, which we don't know how those changes are going to affect the behavior at all. It doesn't mean it's by definition dangerous. It just means we don't know what it's going to do, but it's not going to necessarily behave like it was, developed to behave.
And I mean, that makes sense considering that ibuprofen example that you just gave us. The addition of a single methyl group, which for those that don't know is just a carbon atom attached to 3 hydrogen atoms. So just 4 additional atoms attached to the molecule. If that made such a big difference to how a drug was performing in the body, then what's a big old vitamin doing? Like, that's massive in comparison to a methyl group, just as far as like molecular scale goes.
Yeah. And I mean, the structure function relationships are different for different types of modalities. So for a small molecule, that single methyl group is probably going to have a more profound effect than a methyl group would have on like an antibody or a peptide. But nonetheless, you're talking about a peptide and a vitamin. So it's, it's more. So yeah, I don't want people to be getting, contaminated peptides off of the whatever website they're ordering from and getting these horrific infections. But I also am nervous for people who think that these changes in regulation now mean that they're approved drugs. Because I've encountered a number of people in like the fitness wellness community who are like, doesn't this mean they're now FDA approved? I'm like, no, it's a loophole. They're actually like, I forget what, exempt. Mm-hmm. They're exempt.
So we have to talk about that a little bit because there's no way people don't know, right? All these things that you said, the risks, the lack of testing, the lack of data, but yet they're being marketed so aggressively. Like how do we reconcile those two things?
That marketing is actually a big part of why I've started making videos about this. I teach about drug development and drug discovery. I enjoy teaching. I like making scientific communication content, and peptides was never what I planned to talk about. But the more I see this, what I consider sort of predatory marketing, the more I feel like I need to say something because you have people like my mom, who's a retired scientist, is like, oh, I saw this thing. Like, what do you think about this? And it wasn't a peptide, but it was some other supplement being targeted to the like menopause community. Yeah. It's like, no, it's marketing. Marketing is really powerful. There's psychological basis behind marketing and it sounds really appealing. If you're someone who's going through, let's say I used perimenopause as my example most recently, just because that's the one I can relate to. But, you're tired, like you have all these symptoms and someone tells you, oh, you can fix all of that and you can feel like you're 10 years younger and you just have to like pay $300 a month or whatever and inject these things at home. It sounds great until you actually look up what they're talking about. And if you go and look at the papers they reference, if they even provide the references, which they frequently don't, and they're all data from cells or rats. And, we all know that animal studies do not always translate into humans. Mm-hmm. That's why we have human clinical trials and we don't approve drugs based on mouse data.
So if you were a betting woman, what do you see happening? I mean, this is booming, all eyes on peptides right now. What do you expect will happen in the next few months, next few years? Like, what are the next steps? What do you think people can expect?
I think you're going to see more mainstream availability if the changes that are being proposed actually happen. I think you'll see continued marketing and promotion of peptides. I think the risk of getting something that's dangerous because it's contaminated or it isn't what it says it is will hopefully go down. I mean, that's sort of part of the point of this. I don't know what the threshold of evidence will be or what the rules are because it will be a prescription from a doctor. But if you think about that, if a doctor decides, oh, let me prescribe this peptide to you, and they know that peptide is not an FDA-approved drug, right? They have malpractice insurance. Right. So what happens if something bad happens? Is it the doctor who's liable? Do you have to sign a waiver so that you're liable? Is the compounding pharmacy liable? Like, I don't know how all of that works. I imagine all of the malpractice insurance companies are probably also having similar discussions because they have to figure out how they're going to deal with it. Maybe there's a process. It's not something that I have expertise in because I am not a physician. I think if there, as someone who works in the space, I have to believe that if there was really compelling data in animals and in in animals and in cells and other in vitro systems, the pharmaceutical company would be investing to start developing these things. Like, they're not going to leave money on the table, right? And I know people think that pharma doesn't have our best interests in mind and that they are very skeptical. And there's a lot of people who feel neglected by the medical infrastructure that we have and our health insurance company. I mean, I get it. I'm currently in an argument with an urgent care about a $400 charge for an x-ray for my daughter because the tax ID on their bill is for a different organization that would be considered out of network, even though this urgent care is in network. So it's like, that's, something I'm dealing with. And that's a very minor thing. Like, there's so many issues and people just feel very, skeptical of the medical world, which I completely understand. I get why people want to use these. And like, I don't judge. If you want to do this, go ahead. It's more like when you're selling it to someone else and you're not giving them the whole picture, that's where I have an issue.
I think it's safe to say, Leigh, that you would not inject peptides at this point in time. I would not,
No.
based on the available data. But to
I
your
would
point,
not, but I'm also afraid of needles.
Yeah, well, that's right. Oh my God,
You know
Sarah.
what? Once you've had children, those fears go away. Like putting the hospital gown on, the needles, all those things, like do whatever you want to do.
Exactly, yeah.
After 30 hours of labor with my first one, I'm like, I don't care.
But yeah, I mean, I think it's safe to say, though, that if there were data suddenly, right, if we did do all the necessary preclinical trials and then clinical trials, and if the data supported, that these are safe and effective, we would be behind them, right? I think it's now
a lot of the pathways that they're promoting and the animal data that they talk about, things like, tendon healing and like, improving wound healing or, recovery after injury. They're all things that sound really appealing. I mean, if you have some type of orthopedic issue that's non-surgical and there was something you could inject that would make you get better way faster, like that sounds awesome. But I'm not going to do that unless I know it's been evaluated, because the flip side of this is that the pathways that are being called out, the molecular pathways, involve cell proliferation, cell migration, growth of blood vessels, which is angiogenesis. These are all pathways that are often dysregulated in cancer. So not saying wellness peptides are going to give you cancer. It's just those particular pathways are things that I would be very concerned about inducing in an unregulated way in my body without any data. Certainly there are people who are using these chronically, which makes me really nervous. Short term, maybe that's less risky. But these are also not things that are going to show up as an adverse event in a week. These are things that are like long-term risk.
Can I just try to restate? I mean, I can't restate what you just said. It was so brilliantly stated. But if I understand correctly, or correct me, that the peptides, they lead to some cellular changes or changes in the body that have been associated with cancer. I mean, again, we're not saying that there's any data showing the linkage between peptides and cancer, but theoretically, there's some sort of potential link.
Yeah. And there are also peptides that are inducing like growth hormone and things like that. So, I mean, Sarah, you teach biochemistry. Yep. Do you want to induce cell migration or proliferation or angiogenesis in your body?
Nope, because cancer is basically uncontrolled cell growth. And if you're messing with the biochemical machinery behind what causes cells to grow, then you could potentially be kicking off a cascade that you cannot, sometimes you can't put the toothpaste back in the tube.
Yeah, exactly. And there may be no risk. We don't know. We don't have the data, right? And with that type of biology, I would want to see data. And if you develop drugs that are in that area, there are very specific evaluations you have to do, like a 2-year study in animals to show that you're not seeing any, tumor progression or tumor growth, or in animals that have tumors, make sure it's not going faster or exacerbating. I can't tell you the exact studies because that is not my area, but there's definitely specific evaluations that need to be done if you're going to promote those types of pathways.
Wow.
When you develop a drug.
Well, Leigh, as we wrap up here, is there anything that we didn't discuss that you feel we'd be remiss if we didn't cover in this episode?
Yeah, there's one aspect of preclinical drug development that I think I don't see people talk about the preclinical space a lot. And since that's where I work, I'm obviously biased in that sense. Everyone talks about the phase 1, 2, and 3 trials, and then you get all of this skepticism about the FDA and pharma and regulatory agencies. Where my concerns stem from actually start even earlier, before you're even involving any of those regulatory agencies. For a lot of these peptides, we don't even know what the molecular target is. So when you're developing a drug, now, not historically, but now we generally do target-based drug discovery. So we say, okay, here's a kinase, which is an enzyme that I identified, and it's really important in cancer. I want to make a kinase inhibitor that will be a drug for this particular cancer. And you go through all those safety studies I mentioned to identify whether that is a safe drug target. Then you start trying to develop your inhibitor. So you understand what you're binding. You actually do studies that show that binding event, how long it happens, show that it happens in cells, show how long, how potently you affect the activity, all the downstream pathways that are modulated. Like, you can trace the entire process from end to end biologically before you ever touch an animal. And that's the space where I work. So the fact that we don't even know the molecular target for a lot of these pathways, we know different pathways peptides, not pathways. We don't know the molecular target. We know some of the pathways they might evoke, but they're also very pleiotropic kind of broad pathways, which are unlikely to be modified by one specific target. So they may also have multiple things that they're affecting. That would be a red flag for me, as well as some of the data that's out there. Like, you see on BPC 157, the data all comes from a really small group of researchers. There's not a wide scientific consensus of data on it.
Right.
You don't see a dose response effect in animals, whereas you give more, you see a larger effect. We also don't have any pharmacokinetic, pharmacodynamic data, which is where you actually give something and then you look at the downstream pathways that are affected. So, even in that preclinical phase, there are big gaps in what you would need to do to even get that permission to go into humans.
We are so
a lot of
far from, sorry, it's just wild to me.
No, it is. Yeah. It's because these are not processes that people understand because we don't talk about them. And it isn't anyone, I didn't know about any of this before I did this job.
Right.
This is stuff that you learn by doing. It's not taught in grad school. It's not taught in med school. Preclinical drug development is something you learn by doing preclinical drug development. And same for clinical and all of the phases. These are such niche areas that you have to really do that job to learn that science.
Wow.
So,
well, Dr. Leigh Baxt, we learned a lot from you. I'm going to turn to Sarah to wrap up, but can you just tell us where folks can follow you?
Oh, yes. I am on Instagram at ScienceMomSciComm. I also collaborate heavily with Unbiased Science, so you'll see me tagged on lots of those and vice versa. I do some Substack as well, also with Unbiased Science, but primarily Instagram.
All right. Amazing.
Thank you so much again for joining us. And thank you all for tuning in. We hope you learned a thing or two. I certainly did. Please join us next week on your trusted source for no nonsense, just science.
If you like the show, please take a moment to rate, review, and subscribe. It really does help the show to grow. Thank you for listening.